Again, I'm going to ask you to describe the digestive process of one of these macros. Okay. For here, protein digestion, if you want to say the mouth, mechanical digestion, fantastic, do it. Okay. So there is a little bit that we start. I mean, you could even go back even further. You could say, I've cooked the food. I've cooked the food. I've chewed the food on the stove, in the mouth. That's fine. But where protein digestion truly begins is really in the stomach from enzymatic standpoint. Okay, we have no acidic environment in the mouth, hopefully. And we don't have any enzymes that are being secreted there within the mouth and the saliva that breaks down proteins at all. That begins here in the stomach with hydrochloric acid. Remember pepsinogen from way back when in chapter 4? Pepsinogen plus HCl, hydrochloric acid, gets me pepsin. Pepsin is the gastric enzyme. Gastric means stomach. Pepsin is going to be that enzyme that is breaking down proteins. Polypeptides broken down into smaller peptide chunks. After these smaller polypeptides, again, in the stomach, hydrochloric acid, pepsin doing their thing. Pyloric sphincter opens up and gets squished out into the small intestine. After these polypeptides enter in that small intestine, pancreas gets in on the action and secretes trypsin and chymotrypsin. Long polypeptides broken down into smaller polypeptides in the stomach. In the intestines, pancreas secretes trypsin and chymotrypsin. These break down these smaller polypeptides into shorter peptide chunks. The intestinal cells will secrete enzymes as well. And let's just keep it really super easy. Dipeptidases and tripeptidases. Dipeptidases, remember ASE means enzyme. Dipeptidases break down dipeptides. Tripeptidases break down tripeptides. Bada boom, bada bing. Trypsin and chymotrypsin break that shorter polypeptide sequence into di and tripeptides. Dipeptides broken down into a single amino acid. Tripeptides broken down into maybe a single amino acid, and ideally we want all individual amino acids, but that can be broken down into 1 and 2. And then hopefully a dipeptidase will work. If not, you can get some absorption of dipeptides, but over time that can lead to some inflammation as well. Just big structures. Okay, big structures, when we absorb them, can cause some potential damage. All right, so I have the stomach with HCl and pepsin, breaking down that long polypeptide sequence into shorter polypeptide sequences. In the small intestine, the pancreas secretes trypsin and chymotrypsin, breaks down those smaller polypeptides into dipeptides and tripeptides. The intestinal cells themselves will release enzymes, dipeptidases, tripeptidases. So as those proteins get closer to the intestinal cell, it secretes those enzymes and we break that peptide bond, and now they're their individual amino acids and I can absorb them. And again, they all enter in the capillary, right? We don't have to go to the lacteal like the lipids did. Enter into that capillary and that little villus, travel to the liver. Liver keeps them, releases the rest back into circulation. From a digestive standpoint, this one's really easy, right? Do I like asking easy short answer or essay questions? Probably not. I know we've only had one exam, but you can probably figure out that I'm that DH. But could I throw a bone and ask this one? Yeah, even though there's just a few moving parts. Okay, stomach, HCl, and pepsin. In the liver, I'm sorry, in the small intestine, I got the pancreas secreting trypsin, chymotrypsin. And then the intestinal cells secreting their enzymes as well. So there's a few moving parts there. So I guess I could potentially ask that. From an absorption standpoint, carbs and proteins are really pretty easy, right? There's not much to it. They enter into the blood vessels, into the capillary, which is the blood vessel, and hepatic portal system to the liver, and then to everywhere beyond. So not a whole heck of a lot of stuff there. Who had a lot of moving parts? Lipids. Yes, exactly. So if you're a gambling person, what would you probably imagine that I'm going to ask on the exam? See how easy this is, guys and girls? Very easy to figure out how the hell I'm going to ask a question after you've had an exam, at least I think. From a transport standpoint, did I go into the mechanism of how carbs and amino acids bind to some of the components within the blood and get traveled around? No, they're just water soluble. They just bind to something within the blood and easily transport it. Who has a lot of moving parts when talking about transporting throughout the body? Lipids, yes, one person's getting it. Okay. And for those I can't see, I know you mouthed those words as well. If there's a lot of moving parts, you can probably guarantee I'm going to potentially ask it as an essay exam question. Are there several of those scattered throughout? Yes. But I'm pretty nice because I tell you, this will make a great potential essay or short answer type question on the exam. Digestion of all these macros. The transportation of these macros. Absorption of these macros. We're all potential good ones. One of them stands out a lot more than the other when it comes to a couple of those aspects like we just talked about. How we utilize these inside of a cell, that makes a good short answer question or essay question. And you're probably thinking, where have we talked about that? We haven't done that one yet. That's in chapter 8. Okay. Where we look at how the cells actually utilize glucose, fatty acids, glycerol, amino acids inside the cell, and how it makes that energy. Okay. Asking one of those is a good short answer question or essay type question on the exam. And as we start to get into that on Wednesday, that will become abundantly clear which one I'm probably going to ask.